Hi, my name is Nikki Williamson, and I'm a third-year PhD student in the graduate group of ecology at UC Davis, as well as an Arctic scholar. My research focuses on how climate change is impacting Arctic ecosystems, more specifically, how vegetation shifts might be affecting large herbivores. One thing that really drives my research is a deep interest in protecting animals that are both ecologically and culturally significant, such as caribou. For thousands of years, these creatures have been central to the cultures and livelihoods of many indigenous communities across the circumpolar north, providing food through subsistence hunting and playing an important role in cultural identity and traditional knowledge. At the same time, many caribou populations are declining. Migratory tundra caribou have declined about 65% over the past few decades, and climate change is rapidly transforming the landscapes that they depend on. Because of this, I'm interested in understanding how environmental change is affecting the quality of food that sustain these animals. So one of the big questions driving my research is, how does climate change alter the nutritional landscape for Arctic herbivores like caribou? And to study this, I conduct field research in Kangerlussuaq, Greenland, where warming temperatures as well as shrub expansion are rapidly changing the vegetation for animals. Caribou don't just respond to how much vegetation is available, they are responding to what's in it. Nitrogen content in plants is especially important because it's strongly linked to protein, which influences reproduction, calf survival, and overall population health. If plant nitrogen changes across the landscape, then those changes can ultimately affect caribou populations. And so the challenge is that measuring plant nitrogen the traditional way is extremely labor intensive, and it only gives you information at a single location, but caribou are moving across huge Arctic landscapes. And so the question then becomes, how do we measure forage quality at the scale that the caribou actually experience it? To address this, I pair multispectral imagery collected with a drone that captures light in several spectral bands with plant samples from the same area. Because plant chemistry affects how leaves reflect light, these signals can be used to estimate plant nitrogen. This allows us to create spatial maps of plant nitrogen, which are essentially maps of forage quality across the landscape. This work is important not only because caribou populations are declining, but also because large herbivores play a major role in shaping ecosystems. They graze, they are responsible for nutrient cycling, they can increase plant diversity and help maintain resilient landscapes, and in some systems, large herbivores may even help buffer against some of the effects of climate change. So conservation of these animals is not only important for the species themselves and the communities that depend on them, but it's also really important for maintaining healthy, resilient ecosystems. Looking ahead, I hope to scale this work up even further by linking drone data to satellite imagery. This could allow me to map plant nitrogen across entire Arctic regions, helping scientists and communities understand how warming-driven shifts in vegetation are affecting caribou in the area. I'm so thankful for my support from the ARCS Fellowship, and I really look forward to sharing some exciting research updates with you soon.